Now we're going to discuss a second challenge to macroevolution. Is there a large pool of junk DNA in which gene evolution can occur? Genes are in at least two categories. There is coding DNA. Those are genes with instructions on how to make protein. Then there's also non-coding DNA or silent DNA, which has been considered to be junk DNA because it doesn't seem to do anything. It does not making protein. That has been the prevailing theory for some years. But there has been increasing evidence that questions the whole idea of junk DNA. This article from the journal Nature is one example. It says, is it the junk that makes us human? Could it be the non-coding DNA that actually defines what it is to be human? A large research project was done looking at the literature on junk DNA with a very interesting finding, and that is, of the total DNA that we have, very simple organisms don't have very much junk DNA, only a very small percentage of junk DNA. As animals get more and more complex, the percentage of junk DNA increases, until in humans, 98% of our DNA is junk DNA. Would you be willing to have 98% of your DNA removed, since it's just junk? I'm not sure we would want to do that. This increase in evidence that there is now junk DNA came to a head in 2012 with completion of something called the ENCODE research project. This was a massively government-supported research project which found that virtually all human DNA is functional. And so we can say that junk DNA is really no longer a useful concept. Instead of being useless, it's actually regulatory DNA that is controlling other genes. And so there does not seem to be a pool of junk DNA to support evolution. The supposed junk DNA controls whether our protein-coding DNA will make a human, a chimpanzee, or a mouse. Now we come to a third challenge to macroevolution, and that is evidence for the tree of life. This diagram shows what we call the tree of life in evolution. The phylogenetic tree that supposedly shows how all these different groups of animals evolved from protozoa and bacteria. But the evidence for this tree of life, or for the common ancestry of all genes and organisms, is facing serious setbacks. The Darwinian understanding of macroevolution requires that all new features arise by a long, gradual series of small changes. And the tree of life is claimed to be a record of this series of very small changes. But there are problems, and one of the biggest ones is what are called orphan genes. And these are genes that seem to just appear with no evolutionary ancestry. In this diagram, you see the letters OG, that stands for orphan genes. And so that squirrel has orphan genes that have no ancestral genes in other mammals. That butterfly, another type of insect that has orphan genes, is honeybees. Honeybees have some genes that tell it how to make honey, and no other insects have those. They just appear all of a sudden as orphan genes. How did that happen? And these are not uncommon. At least 10 to 20% of all genes are orphan genes. There are hundreds of those in humans alone. Some are very important, even being responsible for the large human brain. In summary, these orphan genes are a challenge to evolution. In fact, I would say that they are a deadly challenge to Darwinian macroevolution.